there could be like obvious, you could find it pretty readily, or sometimes you don't find it until you run the genetic testing in that way.
And you're now being recorded, just so you know, and you are now co-host.
Thank you. That's super creepy, Dr. Olson. In the dark there. Okay.
It does look like a really bad horror movie on his end.
I know. I was like, that is terrifying. Okay. So we kind of already jumped in, but I don't know about you all, but I am feeling like this 30 Rock meme right now. You know, what a week, and it's only Wednesday. So go ahead, type in the chat for me. What's your hedgehog number? How are we feeling today? As we get ready to dive deeper into sexual differentiation. These are all horrifying in some way. I'm glad we don't have very many ones because that one terrifies me. All right. So we're all feeling somewhat hedgehoggy today, various stages of terror-inducing, horrifying hedgehogs. We're going to continue talking about sexual differentiation. We're going to go over some different examples of things, and then we'll run through a few case studies in small groups so that we can practice. Okay. As always, feel free to interrupt and ask questions along the way. All right. Sea urchin with teeth. All right, so I know we covered this last time. I just figured we'd start with it since get us all back in thinking about the ways in which things can manifest. So this is androgen insensitivity syndrome. This is a condition where the individual might be XY, but have a genetic mutation on the androgen receptor, which is on the X chromosome. So males who inherit this, they're producing testosterone, they're producing DHT, the gonads do develop as testes, but they're not able to respond to those hormones. So they're going to develop phenotypically as female. And you can see here, the external genitalia looks very feminine, and we even have female secondary sex characteristics because of the low levels of estrogen produced by the adrenal gland is enough to stimulate things. Female is the default, so this is what we're going to go towards whenever there's kind of a like an androgen receptor mutation or anything that we're not able to respond to the male hormones as readily. Okay. Here's another condition. This is congenital adrenal hyperplasia, or CAH. Individuals with this condition are born with enlarged adrenal glands. They also lack a specific enzyme that's needed to produce cortisol. So if you remember from the stress pathways that I know you've talked about already, if there's no cortisol there to provide the negative feedback, what's going to happen is the hypothalamus is going to keep releasing more and more and more CRH. The pituitary is going to respond to that and release more and more and more ACTH. And the adrenals are just going to keep growing bigger and bigger and bigger, but they don't actually release any cortisol. They can't, they don't have the enzyme. So they'll release more and more other hormones from the adrenals. The other hormones in the adrenals are some of the sex hormones, those steroid hormones. So when this happens in females, the bodies are exposed to high levels of androgens and partially masculinized. So this is a condition that affects cortisol, but because of this, we're going to be releasing more sex hormones from the adrenals. And there's that window of time where genetic females can be perturbed by early exposure to these androgens. So what's going to happen here is we have clitoral hypertrophy. So it gets enlarged. It looks larger, almost like phallus-like. And the folds of skin kind of develop halfway between what we would consider feminine and masculine, somewhere between labia and scrotum. So this is one of those conditions that we classically call intersex phenotype, where it's an intermediate phenotype between what we would consider typical male and typical female phenotype. So you might be thinking about treatment. Traditionally, females with, well, all people with this condition can be treated with exogenous cortisol to reduce the ACTH release, but that's not going to undo the fetal masculinization that has occurred in the females in the periphery, like in the genitals. Traditionally, historically, doctors have tried to surgically correct to female in this for people with this condition, because it's easier to surgically correct the genitals to be more feminine from this masculinization. However, it doesn't always work. And the new kind of way of thinking about things is to wait until at least after puberty for the individual to decide how they want their body to be, essentially. Okay. So we were talking all along now about what's going on in the body, kind of more in the periphery. Now we're going to start thinking about what's going on masculinizing, feminizing in the brain. So this is a big, huge, important point. You see the star there at the top. This is the organizational activational hypothesis. This is going to come up again and again and again and again, not just this chapter, but the rest of the chapters you talk about. So the organizational activational hypothesis tells us that behavioral sex differences result from, one, we have differential exposure to hormones in early development, and these differences in the hormones early in development are going to organize the neural circuitry that underlie what we would consider sexually dimorphic behaviors. And then the second part of this is the activational part. There's differential exposure to sex steroid hormones later in life, post-puberty, that activate the neural circuitry that we previously organized. So we have to organize the brain to be male or female, and then we have to activate those pathways post-puberty to activate those behaviors. Okay, so here's this example in rodents. And what you can see here is, so let's do female here. We have XX, development of ovaries, default to female. There's going to be very little brain estradiol, because again, the ovaries aren't really producing any hormones in development. Default is going to be female, so no hormone exposure during development. We're going to organize to female. Then later in life, activational, high levels of estradiol and progesterone will lead to, and in rodents, we typically look at sex behaviors. So this is a position called lordosis that the female rodents will, they kind of arch their back in this way. If they do not get into this position, the male cannot actually have sex with them. It doesn't fit. So this is a sexual dimorphic behavior. Only the female rodents show this position, this lordosis. In the males, so we have XY, the SRY gene is present. We're going to develop testes. We're going to have the testosterone. We're going to go over this again in a minute, but actually testosterone is going to be converted to estradiol, and it's the estradiol that is going to masculinize the brain. So the high presence of estradiol in development is going to masculinize, organize the brain circuitry, so that post-puberty, when we have high levels of testosterone, we will get mounting behavior and we do not get this lordosis position. in the males. Okay. So if we think about sexual differentiation of the brain, it's a little bit backwards. People sometimes get a little thrown by this, but actually it's estrogen that is masculinizing the brain. So testosterone is being produced in the male fetus, right? We have an enzyme called aromatase, which is going to convert it to estrogen. Estrogen is going to masculinize the brain. And when I say that, what do I mean? I'm meaning it's going to create neural pathways that are more sensitive to testosterone. We are going to masculinize the spinal cord centers. So we think about differences in pain sensitivity. That's happening early in development here. The hypothalamus will be masculinized so that it will do pulses of various hormones at different timing. It will also defeminize. It inhibits the development of brain circuitry in the female direction. Okay. So this is all happening at about 6 months of gestation. This is sexual differentiation of the brain. So now if we have females, we don't have testosterone being produced in the fetus. So therefore we won't have any of this happening. Default will be female. The pathways will just naturally go towards the feminization. So hypothalamic differentiation, spinal cord centers, not being sensitive to testosterone later in life, all those things. So question in the chat here. I've heard tofu is full of estrogen and it's not good for females to eat. Have you heard about this? So soy can definitely increase estrogens, and it's not necessarily a bad thing, but it can be people who eat it in extreme amounts, very high amounts, can have what we would consider out of the normal physiological levels of estrogen. They can have larger levels. So that would be the concern there. Okay. So what do you think about this? Does it make sense that it's estrogen that is masculinizing the brain?
I'm a little confused because you had said, like on the last slide, the males, since the estrogen is masculinizing the brain, why don't we see something like, if estrogen is only working in the females, why doesn't that happen? Is it because it's the estrogen converted from testosterone, or that bit confuses me a little?
That's a great question. Perfect transition for us. So you might be thinking very much along the same lines. Why aren't all fetuses therefore exposed to high levels of estrogen? Because moms are all having high levels of estrogen. In fact, in pregnancy, that's one of the highest times you see for estrogen. It's very high. So why is mom's estrogen or the estrogen from the adrenals not enough? It's because testosterone is entering the cells, right? Converting to aromatase here to estrogen, binding to the estrogen receptors and causing the effects. In females, there's something called alpha-fetoprotein. The mothers are producing this alpha-fetoprotein. It binds estrogen, but it does not bind testosterone prenatally. So any estrogen that is being produced by the mom or by the adrenals is going to be captured by this alpha-fetoprotein, making it too large. It cannot cross that barrier. Male fetuses circumvent this barrier by secreting testosterone, which can bypass. It doesn't bind to the alpha-fetoprotein. And so testosterone converts to estrogen. You might be thinking, could that not happen in the females? Could the estrogen convert back to testosterone? It can, but it's very energetically expensive and almost rarely happens, functionally insignificant. Okay.
Is aromatase also present in females?
It is. it is present in females, but not in the fetal, really, development, because the ovaries are not secreting any hormones at this point. No. They're just there. So there's nothing for aromatase to do. But the male gonad in development is producing testosterone, which can be converted. All right. So here's again kind of what that looks like here. We have this organization. Think of that as the sensitive developmental time period where the brain has to be exposed to the estradiol to organize those brain circuitry pathways. Therefore, post-puberty, when the sex steroids are circulating, those hormones can now activate those pathways. And what we're going to get here, I have too much on my screen. Around. Is estradiol the same as estrogen? Yes. Basically, it's the estrogens are, we'll refer to them as a more broad term. Estradiol is a little more specific, and different species might have different structural pathways. But yes, you can use them interchangeably here. Where is that aromatase located? Good question. In the fetus, aromatase is really located solely in, there is some in the scrotum, but in the genital region, like with the testes, but more of it is present in the brain, which is why the testosterone can go into the brain, convert to estradiol there, masculinize those pathways. In the females, there's aromatase in the brain as well, but there's nothing for it to really act on because there's no testosterone. And then later in life, there will be lots of aromatase in the ovaries to convert the testosterone to estradiol. Okay. So this is what I mean by the pulsing the hypothalamus. So you're going to get into this when you start talking about sexual behavior, male, females. But in males, there's testosterone surges, and that causes luteinizing hormone, another hormone from the hypothalamus. And it does it in these pulses. You can see pretty regular pulses. In females, we have the pulses of luteinizing hormone, and then we have surges, and then pulses. That is all set up organizationally in that pre-stage in development. If you take a female rodent in development and you give them testosterone, you're going to masculinize that brain. You only give them the testosterone early in life, in that developmental time period. In adulthood, you're going to see the luteinizing hormone pulses that look masculine. You're not going to get the surges. If you castrate the male during that developmental time period, you don't get the masculinization of the brain, and later in life, you're going to see feminine luteinizing hormone surges. Another behavior is that sexual behavior. So males masculinize the brain, later in life, activate those pathways. We get male sexual behavior. We get the mounting behavior in the rodents here. Females, we don't have the testosterone. We don't masculinize the circuitry through estradiol. Later in life, post-puberty, we get the lordosis position. Same thing. If we take that female in development, give them testosterone during those crucial time periods, later in life, we're going to see the mounting behavior. Males castrate during that critical developmental time period. Later in life, you're going to see the feminine sexual behavior, lordosis. So I see a question in the chat. How would you increase levels of estrogen in women? Is that related to development or generally? Can you just be a little more specific for me, Cassidy?
I was just thinking more generally because if the
Are you thinking about the alpha fetoprotein?
Can you hear me?
I can hear you.
Okay, yes.
So you were thinking about the alpha fetoprotein, like binding?
Yeah, mainly that, because if that kind of turns it into testosterone, then I was confused how.
So the alpha fetoprotein is actually only present when you're pregnant.
Oh, okay.
So generally speaking, typically like you could just give exogenous estrogen, or you can give females testosterone. We have testosterone receptors. Females have testosterone receptors. It's just that more of it will convert to estradiol.
Okay, thank you.
Yeah. And as a reminder, males also have estradiol. Males also have progesterone. They just have it in different levels than females. So it's very easy to fall into this pattern of saying things like, testosterone is the male hormone and estradiol is the female hormone. It's not really accurate. We all have both, really. It's just in what percentages and what are those hormones doing at those key developmental time periods. That's really more of what it is. Okay, so here's a crazy chart. This is on your worksheet too. At first, it might seem a little overwhelming, a lot of yeses and nos here, but let's walk through it, okay? And think about what's happening. We have been able to discover basically all of this about the organizational and activational pathways by doing some very simple experiments, really. Is there a trigger that would cause a female fetus to get too much estradiol and masculinize the brain? Basically, the only way that's going to happen is if that fetus is exposed to androgens. So if the mother is pregnant in that critical time period and is taking testosterone or environmentally exposed to androgens in this way. That's really the only reason why that would happen. Or the congenital adrenal hyperplasia, where the sex steroids being released from the adrenals are much greater levels than typical. All right, so let's look at this pathway here. Let's work with the male pathway first, okay? Or no, let's do the female. Female's easier in this case. So we have the females. Do we remove the gonads before day 10? No, we don't have to do any of that because again, female default. So now we can either inject with testosterone or not inject with testosterone. So let's say we don't inject with testosterone. We're sticking with the normal feminine pathway. Do we remove the gonads in adulthood? Yes, we do this so we can experimentally control what they're going to receive in adulthood. So think of that's injecting with testosterone as the organizational. Injecting with testosterone in adulthood is going to be the activational. So if we don't inject with testosterone, that's typical female pathway. Are we going to get male typical behaviors? No, we're going to get female typical behaviors. We do not inject with testosterone. We do not organize the brain to be masculine. We do give testosterone in adulthood. No, we do not get male typical behavior. The reason we don't get typical male behavior in this case, even though we're giving testosterone to these adult rats, is because we have not organized the brain to respond to testosterone that way. You have to have both. So now here we're going to inject with testosterone in these female rodent fetuses, critical time period developmentally. So we are going to organize the brain circuitry to be male. If we don't give testosterone, if we don't activate the pathways, we're still going to get the female typical behavior. If we do give testosterone, though, we will get the male typical behavior because we have organized and activated. How are we feeling on that? Okay. See a couple thumbs up. Thank you. All right, let's walk through the male pathway. We don't remove the gonads, so we're going to keep the testes. We don't inject with testosterone. We don't give testosterone in adulthood, so we've activated the pathway, yes, because we have the normal gonads, but we don't activate it in adulthood, so no male typical behavior. We have organized the brain. We give testosterone in adulthood, we activate it. Yes, male typical behavior. We inject with testosterone. This is really just going above and beyond. You don't need to because we haven't removed the gonads. So they're just getting a lot of testosterone here. So we are organizing the brain to be male. We do not activate it. We do not get the male typical behavior. We do activate it in adulthood. We do get the male typical behavior. So now we remove the gonads. So now it's really about the hormones. Remember those three kind of things we can do to see if a behavior is dependent on a hormone. We can remove the hormone and see if the behavior disappears. We can put it back in, see if it comes back. That's all we're doing here. So we don't inject with testosterone. So essentially in this case, in this pathway, what we're doing, we are simply going down the female default pathway. We are not activating the brain to be male. We don't give testosterone. We don't get male behavior. We are not activating the brain. We do give testosterone in adulthood. We still don't get the male typical behavior. And the reason is we haven't organized the brain. We follow this yes pathway where we are going to organize the brain to be masculine. We don't give testosterone in adulthood. We haven't activated the pathway, so no male typical behavior. Yes, we have organized the brain to be masculine. Yes, we are activating those pathways. Yes, we get the male typical behavior. Okay. So I know it's a crazy flowchart there, but what you want to be able to do is make sure you can follow the logic and get to whether or not there would be male typical behavior. To get a yes for male typical behavior, we have to both organize the brain to be masculine by that exposure to estrogen in the critical developmental time period. And then we have to activate those pathways post-puberty with the testosterone. How are we feeling? Thumbs up, thumbs down? Awesome. Okay. So this actually creates a little bit of an interesting scenario for us. This is only everything I'm going to talk about right now, only for fetuses that are basically in litters where they're exchanging the sex hormones via the placental circulation. So this is really not humans, but other mammals and species that have litters, right? So we can look at the position in the uterus of the fetus. All right, so here we have male, female, male, 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 female, female, female. All right. So females, right here, this one, that develop in between two male fetuses, so 2M females, are exposed to much higher levels of testosterone than siblings that may be next to one or no males. So what we actually see is these 2M females, these ones that are exposed to more testosterone just based on their placement in the uterus, they have more masculinized genitals, they have shorter reproductive cycles, and they are found to be less attractive to males in adulthood. Males find them less attractive. We also see the converse here. Males that develop next to two females often have smaller seminal vesicles, are less aggressive than 0F males because males near other males are going to be exposed to just more testosterone too. So it's really important about how we are organizing everything early in life. So this is just a really interesting example of how these sex hormones are working. Questions, thoughts on this? Okay. So let's kind of re-summarize where we are and how we got here. So sexual differentiation is the process. Sexual determination is that one step, okay? The establishment of the chromosomal sex at fertilization, development of the gonad. So the whole process starts, we have the genes. It's the SRY gene, which is normally on the Y chromosome. If we have the SRY gene, then what's going to happen? We're going to develop testes as the male gonad. The testes are then going to release anti-Mullerian hormone and testosterone. Anti-Mullerian hormone is going to inhibit the Mullerian ducts. Testosterone is going to virilize the Wolffian ducts. We're going to get all the male accessory sex organs. Testosterone is going to convert to DHT and masculinize the external genitalia. Testosterone is going to convert to estradiol in the brain and masculinize the brain circuitry there. So all the steps to become male require these additional steps. Female is the default. So if we don't have something happening or we don't have a hormone, we're going to default to female. So if the SRY gene is not present, if it's XX, then the determination is female and everything is going to differentiate female from there. We won't be releasing testosterone from the ovaries. We aren't releasing anti-Mullerian hormone, so therefore we're going to continue the Mullerian ducts and the female accessory sex organs. There's no testosterone or DHT to masculinize the external genitalia, so therefore feminine external genitalia. There is no testosterone to convert to estradiol in the brain, so therefore feminize the pathways and organize them in the brain. All right. Here's kind of another way of thinking about this, all the different things testosterone is doing here. Testosterone is going to be acting to differentiate the male internal organs. This is the virilization of the Wolffian ducts, also secondary sex characteristics in adulthood. Testosterone is going to convert via 5-alpha reductase to DHT. That differentiates the male external genitalia, forms the scrotum and the genital penis. Testosterone is going to convert via aromatase to estradiol and differentiate the male hypothalamus and other neural circuitry in the brain. Okay. So differentiation of the brain, male versus female. Some of the areas are the preoptic area. This is that area that's really responsible for sexual behavior. You're going to talk a lot more about it later. The gonadotropin secretion pattern in the hypothalamus, the spinal cord and pain pathways are all going to differentiate. Differentiation of the brain is really induced by testosterone converting to estradiol in that critical period. The female patterns are again predetermined. They are the default. All right. So why is it inappropriate to call androgens the male hormone and estrogens the female hormones? This is on your sheet, but let's make sure we're all on the same page.
Because both sexes have both?
Both sexes have both. We both have testosterone. We both have estrogens. Okay. What else? Any other reasons we can think of?
Estradiol masculinizes. the
Yeah. Estradiol is actually what is masculinizing the brain. How could that be a feminine hormone if it's masculinizing the brain? So it's not quite as straightforward as maybe sometimes people try and make it. Okay, so let's think about the subtlety in there. Okay, I am now going to have us work through some scenarios. These are the ones that are on your worksheet, so you have them there. But I also put them in a Jamboard here for you. Okay. So what I'm going to do is put you in some breakout rooms, and let's see, we'll do 6 breakout rooms so that each group can really focus on one to start. So whatever your breakout room is, do that slide. So if you're breakout room 1, you'll do slide 1. If you're breakout room 2, you'll do slide 2. Got it? And then what we will do is, I want to make sure. I want you to go ahead, and you can do this on your worksheet. You can do it in the Jamboard. You can do it just on a other piece of paper, whatever you want. Basically, I want you to think about the scenario I give you and whether or not the gonads are going to be masculine or feminine. So will a testes develop or will ovaries develop? Then what's going to happen to the Mullerian or Wolffian ducts? Which ones will develop? External genitalia, is that going to be masculine or feminine? Is it going to develop into a penis and scrotum, or is it going to develop into upper vagina, labia, clitoris? And then the brain, are the brain pathways going to be masculinized or feminized? All right. Are we all clear on that? Know what you're going to do? When you've done one scenario, go ahead and move on to the next one, and we'll give you let's do about 15 minutes to work through as many of the scenarios as you can, and then we'll come back together and go through them. It's okay if you get confused at any point. You can call for help, and I can jump in to help your group, or you can just wait and we'll all come back together. Okay? All right, I'm opening the rooms. Melina, Mustafa, are you all okay? You need any help getting to a breakout room?
Can you resend it? I think I just misclicked.
No problem. Oh, you got it. Eric, if you're there, you can probably stop recording. All right, everybody should be back now. So I thought we could go through these cases all together.
Yes, please.
Yeah, got
How
a little
was
crumbled.
Yeah.
Did you have lots of debate?
Out loud, though, it helps.
Yeah, I was in the first group. As you can see, we didn't write anything.
That's
we were
okay.
we were trying to figure out, we were following the flow charts and kind of talking about, what would happen. And we were trying to figure out if it would revert to female or if it would still, phenotypically be male, but just without testes. So we were just trying to figure it out.
Yeah. No, so these are meant to be thinkers. These are meant to make you really think about this material and how it's going to act. So we can think of each step kind of independently as we go through. Okay. So we have an XY individual in which testes fail to develop. Okay. So the testes do not develop for whatever reason. So gonads, what's going to happen? We already told us no testes. So probably no ovaries either in this case because of what's going on. So just no gonads, period.
Okay.
Okay. So Mullerian ducts versus Wolffian ducts. The question we want to ask on this level is always going to be two parts. One, is there anti-Mullerian hormone? And then two, is there testosterone? There's no testes, so therefore there's no anti-Mullerian hormone and there's no testosterone being put out. So therefore the default is going to be the Mullerian duct develops. We get to the external genitalia. The question we want to ask ourselves is there DHT present or not? There's no testosterone being produced because there's no testes, so it can't convert to DHT. So therefore, external genitalia will default to female. For brain being masculine or feminized, we want to think, is there testosterone present that can convert to estradiol? There isn't because the gonads are not releasing it. So therefore, we're going to go phenotypically female for the brain. How do we do? So let me throw this one out that was not on there. What if we had an XY individual and only one testy failed to develop? Now what's going to happen?
Will they develop male normally because they still have that testosterone being produced by the testes?
Exactly. A single testy is enough to produce those androgens. So we're going to have the one testy, we're going to get the Wolffian ducts. We'll have anti-Mullerian hormone from the one, so we won't have the Mullerian ducts. The external genitalia will still masculinize and the brain would still masculinize as well. Okay, let's go to your next one. We have an XY individual in which androgen receptors are absent. All right. Maybe someone from group 2. What did you all think?
So we had a little bit of a hard time because we were trying to figure out, I think probably everybody had a hard time because it's just so many different
You were meant
things.
to have a hard time with it, but that's why.
Yeah, that's what I definitely thought. So the way that I looked at it was on slide 42, there's the overview of somatic sexual development. And it just goes through all of those different things. If androgens are not present, then you've said, it defaults to female. So there would not be testes forming. And then the Wolffian duct, because of the lack of androgens, would not form either. So then the Mullerian duct does form, and then external genitalia would be female, and then the brain would be female. That's where I thought it was, but I could be totally off.
It's okay. Thank you for putting it out there and making some guesses. So the first thing we will always want to do is let's just start with the gonads. Okay. what gonads are going to develop? The question you want to be asking yourself is the SRY gene there? We have an XY individual. So is the SRY gene there? Yes, because I didn't tell you this was a reversed scenario. So we have an XY individual, they have the SRY gene, therefore the gonads will develop as testes. Yes, we're going to get testes. Now the testes are what are going to produce the androgens. The testes will start releasing testosterone and anti-Mullerian hormone. So the testes are still producing anti-Mullerian hormone. Anti-Mullerian hormone is not an androgen. So it can still act and can still make the Mullerian ducts go away. However, testosterone is going to be produced, but there's nowhere for it to act. It can't bind to those receptors. There are no receptors. So therefore, we can't masculinize the Wolffian ducts. So essentially, we're going to get no sexual organs internally. So let's go to external genitalia. We have testosterone being produced. We have DHT being produced. But again, no receptors. No receptor, no effect. So therefore, the external genitalia is going to develop
As
Feminine?
female.
Email?
female. Yes. Okay, so we have testes. We're going to have no internal sexual organs. We're going to have female external genitalia. And we have the testosterone being produced. It can convert to estradiol, so it could masculinize the brain. So this individual, we're going to have a masculinized brain, external female genitalia, no internal sex organs. So this person would be sterile. And they have testes and have XY.
Wait, so I have a question. If they have testes, where are the testes? If they're not, if there's a female external genitalia, then where do the testes go?
So this is what we would call undescended. So they're internal, similar to like ovaries.
Okay.
They just haven't dropped down into a scrotum because there is no scrotum being developed here.
Perfect. Thank you. Looks
Yeah.
like I was so off, but you know.
But you know what? They were all, but your logic flowed from where your logic was. So you're thinking about it. That's all we can ask you to do.
Exactly.
So I want you to always start on these scenarios because I can tell you, you're going to get a scenario like this on the quiz. Okay. So always start with what gonads will be present. And that'll tell you what hormones are going to be present, which you can then follow down from there.
I have a question for you on that one with basically the androgen insensitivity. I know that humans that have this largely end up very much so presenting and identifying with female.
Mhm.
So is there a chance that the brain doesn't get masculinized, or does the quote unquote masculinization of the brain not affect humans the same way it would other animals?
No, so the brain is, when we say masculinized, we're talking really about the lateral hypothalamus. We're talking about like pain pathways and like some of those structures and things. There is some evidence coming out that this is also partially affecting sexual behavior and like sexual orientation later in life too, in the sense that areas of the brain can be altered. Oh, you're throwing out some great stuff there. Oh, that's Dr. Olson. Okay. So yeah, the brain would probably still be masculinized. It probably wouldn't be as masculinized in this case as a typical scenario. What you typically see with actually androgen insensitivity syndrome is these individuals identify as female and, you know, externally they look female, they present as female, but Many of them consider themselves to be lesbian or bisexual in nature and typically show more masculinized behaviors as well. All right, let's go through another scenario. We have an XY individual and the anti-Mullerian hormone is absent. So whatever reason, we're not producing the anti-Mullerian hormone. All right, so let's do it kind of all together here. XY individual, so what will the gonads be? Testes or ovaries?
Testes.
Testes. Good. All right. Mullerian duct or Wolffian duct?
Mullerian?
Wolffian.
Wolffian, because it's still producing AMH. No, it's absent, my bad.
So this is actually a case we're going to get both. There's no anti-Mullerian hormone, so we don't make the Mullerian ducts disappear. The Mullerian ducts will develop. There is testosterone. We didn't say anything about testosterone, so the Wolffian ducts will also start to develop. This is going to create, again, a scenario in which we have sterility. Okay, external genitalia, male or female?
Would it be male?
Yeah, because we still have testosterone. Testosterone will convert to DHT. DHT masculinizes the genitals. And then the brain, masculinized or feminized?
I want to say both. Just because we have both the Mullerian and the Wolffian.
So what masculinizes the brain?
It's the testosterone coming from the testes, right?
Mhm.
So, which are present. So it'd be
So we already have that. So we're going to get the masculinization of the brain. Okay. So we can't really, the feminization is more default. That's going to be absent of the testosterone. So we can't convert to the estradiol. So here we have testes. We're going to basically have both developing Mullerian and Wolffian ducts. We're going to have the external genitalia become a penis and scrotum, and the brain is going to become masculinized.
So are we also saying that the brain does not become masculinized unless there is sufficient testosterone from the testes produced?
Yes.
And the testosterone that's present in a XX chromosomal female is not enough ever.
The testosterone that it's produced in development by the adrenals is so tiny that it's not enough to masculinize the brain. Oop. All right, I just threw this one out here. We have an XX individual in which both ovaries fail to develop. So we wouldn't have ovaries or testes. Because of that, we're not releasing any hormones at all. So from there, everything is going to default female still. Mullerian duct will develop, female external genitalia, feminized brain. Does that one track? Was that one a simpler one?
Yes.
Okay. See, the females are so easy now. It's these males that are making it complicated. All right. We have an XY individual in which the enzyme 5-alpha reductase is absent. So what does 5-alpha reductase do?
It converts testosterone to DHT.
Good. Okay. So we are unable to produce DHT is basically what this is telling you. So XY individual, are we going to have testes or ovaries?
Testes.
Testes. Good. Are we going to have Mullerian or Wolffian ducts? We have testes. The testes are by all means producing normal hormones.
Wolfarian.
We're going to get the Wolffian ducts because we have the testosterone to virilize the Wolffian ducts. We can assume anti-Mullerian hormone is still being produced. So we will get rid of the Mullerian ducts. All right, external genitalia, male or female?
Male.
Male.
Yes, Mustafa, female. Why?
Because there's no dihydrotestosterone.
Yep, there's no DHT. We can't convert. We're making testosterone, but what masculinizes the external genitalia is actually the DHT, converting testosterone to DHT. If we don't have the enzyme, we can't make DHT. Therefore, the external genitalia is going to be female. And how about the brain, masculine or feminine?
Feminine?
Would it be masculine?
It's one or the other, right? It's going to be masculine in this case because we still have testosterone, and testosterone can still convert in this scenario to estradiol. Okay. So I realized on your chart and then what I gave you basically two of the same cases at one point. I did the same one about the androgen or anti-Mullerian hormone again. So here's a new one for us in our last minutes to think about all together. We have an XX individual. And this XX individual is going to be exposed to high levels of androgens at 5 to 6 months gestation. So let's do it all together. XX, so are we going to have ovaries or testes?
Ovaries.
Ovaries, okay. So we have ovaries. Are we going to have Wolffian ducts or Mullerian ducts?
Mullerian ducts.
Mullerian ducts, because we don't have, and if you remember, this is all happening, this development at like 6 weeks gestation, this part. Okay, so we're gonna get the Wolffian ducts will recede. There's no testosterone, and the Mullerian ducts develop. All right, external genitalia, male or female?
Female.
Female, yeah. Brain, male or female?
Female.
So this is
male because
where
of higher levels of
the androgens are going to come into play. We're going to get a masculinized brain. And while we're going to have female external genitalia, there might be enough to somewhat masculinize that female genitalia. It might just, we might see it in terms of like an enlarged clitoris, like clitoris hypertrophy. So it would still be visibly female, but it might be a little bit on the spectrum more toward male because of those androgen exposures. All right. How do you feel after working through some of those cases?
Definitely feel like I need to do a few more to like fully understand them, but better.
Yeah, that
So
helps.
always start in order, right? The first question you're going to ask yourself is, what are the gonads? Now, what hormones are being produced from that gonads will drive everything else. Okay. So I would suggest doing that chart maybe again, or coming up with some other scenarios in your head about what might happen at various time points. Okay. Okay. All right. So that's it. We're done for the day. There was a lot there. As you're going through things, if you start going through scenarios and you find yourselves getting confused, feel free to email me. I will make sure Dr. Olson puts it in my email in an announcement, but I'll just put it in the chat for you too. I'm happy to answer questions about any of this stuff as you keep going. I also hope this unit gave you some excellent food for thought as you think about your first paper that you're going to be writing for Dr. Olson about who is male and who is female. How do we determine that? What's the best way to do that? If he hasn't said it already, I'll say it for you. There is no right or wrong answer. It's just trying to provide some evidence for whatever your choice might be. All right. Well, class is over. So Dr. Olson, I think you should probably stop recording at this point. And yeah, hopefully you don't all agree. And I'll stick around for a few minutes in case there's any questions. But I hope you have a great rest of your week.